Hello everyone, welcome to EPG Pathshala. I am Dr. Veda Krishnan. I am a scientist in the Division of Biochemistry, Indian Agriculture Research Institute, that is IARI, New Delhi, India. Today I am going to talk about the overview of enzyme compartmentalization in cells and tissues from the paper Protein Biochemistry and Enzymology. Enzymes. Well, probably you have heard tons and tons of time this word enzyme. In the simplest way, we can say these are biocatalysts, mostly of proteinaceous origin. As you all know, a cascade of metabolic reactions are taking place in any living system which are catalyzed and well regulated by these enzymes. It's intriguing to understand the complexity of living system is maintained through compartmentalization and how they are linked together by enzyme. So in this module, let's discuss on the need of enzyme compartmentalization in living system. Being so important, what are the existing methods for studying enzyme compartmentalization? And finally, briefly on few pathways where enzyme compartmentalization is used as a mode of regulation. Enzymes catalyze various metabolic pathway reaction by proper inter and intracellular compartmentalization. To play this role, there are certain accessory molecules which assist enzyme. That includes energy sources like ATP or GTP and various metabolic precursors like cofactors or coenzyme, or in some case certain intermediates. For compartmentalization of enzyme, cell is solely depending on the nucleus, the master organelle, and ribosome, the protein factories. A given type of enzyme may exist in various isoforms based on the compartment where to act or alter their location within the cell under different physiological conditions like abiotic stress. It is surprising to understand how cell or tissue compartmentalizes its metabolic need. Being so important, various traditional and modern methods have been used to unravel and to understand the localization and regulation aspects of cell compartmentation. The various methods used to study compartmentation are cell fractionation, immunohistochemistry, proteome analysis by mass spectroscopy, fluorescent protein tagging and other in vivo imaging, computational or in silico approaches. Let's briefly discuss more on these. The most traditional method to study cell compartmentalization is cell fractionation, where cells from tissues as well as organelles from cells can be separated by gentle disruption in an osmotically stable medium. This crude tissue homogenate or cell lysate can further fractionate by differential or density gradient centrifugation using various gradients like sucrose, percol, fecol and so on. Each fraction can then have collected and aside for particular enzyme activities. For each organelle, there are certain marker enzymes already well studied and commercially available, like DNA polymerase for nucleus, catalase for peroxisome, and so on. If required, latency measurement and protease protection assays can also be performed in each fractions. Separate organelle fraction can be further purified for downstream assays. Hence, here each compartments are separated based on marker enzymes. So, what are these marker enzymes? Any enzyme which is confined to a particular organelle, cell or a cellular component is called a marker enzyme. Being specific, their detection indicates the presence of source. Marker enzymes are also used as cell biomarkers to characterize a particular cell type, or even sometimes as biomarkers for detecting various disease conditions. Example, succinate dehydrogenase for mitochondria, acid phosphatase for lysosome, glyoxysomes by glyoxylate reductase enzyme. A detailed list is included as table 1 in the text document. This illustration shows cellular compartmentalization by differential centrifugation. The major limitation of cell fractionation technique is that prior knowledge on enzyme localization is required, and in case if it is cytosolic, it can only be referred in a negative way, that is, if it does not co-localize to any non-organelle or membrane compartment. The second technique most commonly used in enzyme compartmentalization studies is immunohistochemistry. As the word immuno shows that antibodies are involved, that means this is a much more specific method. So what is immunohistochemistry? It is a histology-based staining method used to study compartmentalization using specific antibodies. Antibodies are specific and raised against particular proteins or enzyme which bind specifically to the tissue sections and then visualized by light or electron microscopy after labeling with various tagging molecules like fluorescent, luminescent, radioactive or enzymatic in nature. This technique is mostly used for those proteins which do not have enzymatic activity, especially transporters or unknown protein. Histochemistry can also be used to locate non-protein targets such as cell wall polysaccharides and amino acids. This technique can overcome the limitations of cell fractionation of cytosolic proteins. This will also help to study on enzymes which compartmentalize in like intracellular structures like ER, Golgi, etc., which are too fragile or too low in abundance. Coming to the limitation, the need for identifying specific antibodies for each target of interest is expensive and time consuming. Occasionally, weak cross reaction with certain abundant proteins like Rubisco in plant system can lead to misinterpretation of the result. Another major drawback is antibodies depend on structure-based interaction, the epitope-paratope binding. So it also cannot differentiate between native and inactive form of the target protein like unprocessed or partially degraded protein. Thus, immunohistochemistry only reveals the presence of the target enzyme in that location rather than its activity. The two methods discussed, fractionation and immunostaining, are low throughput approaches with many limitations for investigating metabolic compartmentalization. In the last few decades, targeted mass spectrometry combined with classical cell fractionation techniques helped the high throughput cataloging of proteins of various organisms, cell compartment as well as various physiological conditions. As in this figure, this method catalogs, enlists the protein based on the database available and predict its location as well as function. Such whole proteome characterization have not only confirmed the localization of various proteins identified by previous studies, but also capable to reveal novel function to already known or to novel proteins. The major limitation of this technique is the reliability of the proteome result solely depend on the purity of the organelle preparation and the level of cross-contamination by proteins from other compartment. It is also limited by the dynamic range, so very difficult to identify peptide signals from rare proteins. Mass spectroscopic methods cannot distinguish between active and inactive proteins, so that is also a problem with this technique. It is challenging to identify hydrophobic proteins due to the difficulty in preparing highly purified membrane preparations like plasma lemma, ER Golgi membrane proteins, etc. These existing limitations of spectroscopy can be tackled using isotope labeling and using laser microdissection capture microscopy. The existing error chronicity due to microscopy, fractionation and spectroscopy based assays is tackled by a new recent technique based on fluorescent tagging and live imaging. This can be visualized by state-of-art fluorescent or confocal microscopy of the process samples as well as you can see in the figure, in vivo live imaging. Fusion protein construct using GFP, that is green fluorescent protein, YFP, yellow fluorescent protein, etc. has been initially used for pilot studies using transient and stable expression system. These fluorescent molecules are called affinity tags. Let's see what are the major characteristics of these tags. Signal sequences or leader peptides are used to accurately target the fluorescent construct to the locations like plastids, mitochondria, and so on. It is crucial to decide where to attach the fluorescent tag to the target protein, either the C-terminal or the N-terminal, because abnormally high level of protein expression can lead to artifactual compartmentalization. Due to the presence of signal peptide sequences for targeting plasmids and mitochondria on the N-terminal, the tagging should be avoided at this end. So what about C-terminal? C-terminal also contain important targeting information such as endoplasmic reticulum retention signals. So thus, by minimizing the disruption of targeting signals at both terminus, default strategy is to insert fluorescent tag about 10 amino acid upstream to C-terminus, flanked by 6 to 9 amino acid, which is glycine and alanine rich peptide for maximum integrity. NMR, nuclear magnetic resonance spectroscopy, is recently used to determine the intracellular distribution of inorganic orthophosphate and can detect changes in energy or pH of living cells. It is used for measuring flux rate of sucrose or water through phloem and xylem in plant systems. In vivo NMR is poor in sensitivity and can only detect metabolites that are present in millimolar concentrations. A more accurate imaging approach is PET, positron emission tomography, which detects short-lived isotopes like carbon-11 to label sugars and polyphenols. Nevertheless, it is very expensive and hence found very little in application. A holistic approach of various proteomic platforms from fractionation to PET or fluorescent protein labeling studies have revealed the target location for over thousands of proteins in various plants. For example, 4,000 proteins in Arabidopsis thaliana with more than 24,000 proteins with unknown locations. To tackle this huge problem and for annotating function to these unknown proteins, certain in silico computational approaches have been developed based on primary protein sequence. The major softwares are TargetP, Predator, IPSort, Sublog, and many more. Most programs aims to identify the C or the N terminus or signal peptide sequence for accurate location analysis. So far, we discussed on techniques. Now let's briefly discuss on the role of compartmentalization in various pathways. Over the last 4 decades, intense painstaking research on various platforms and techniques were used to understand the role of compartmentation in plant metabolism. Most of the primary metabolic pathways and their location have been well explored and mapped along with few novel metabolic pathways. This proteomics paved a way towards local isomics, which ended with interactomics. Let's briefly touch upon three important pathways. To start with, let's discuss on glycolysis, the very first step of glucose breakdown for energy. Cell compartmentalization studies focused on glycolytic intermediates explored from 1970s to 1980s, along with fatty acids, nucleotides and other secondary metabolism. A striking discovery of the existence of glycolysis and oxidative pentose phosphate pathway in duplicate manner both in cytosol and plastid stroma revealed how energy and carbon skeleton used up in both compartments. The location of glycolytic enzyme to be cytosolic or closely associated to outer mitochondrial membrane is also a result of compartmentalization studies. Several glycolytic enzymes act as a metabolic sensor and transcription factors are found in nucleus as well as cytosol, explains the regulatory role of these enzymes also. Coming to another pathway which is very important, glyoxylate pathway, where lipids convert to sugar in oil seeds. This is a tripartite pathway involving peroxisome, cytosol and mitochondria. Extensive studies done in castor bean seedling solved the existing questions like how carbon is transported between these three organelles, what is the vital role of malate dehydrogenase enzyme, and many more. It was interesting to observe that high NADH-NAD+ ratio built up in the peroxisome arising due to beta oxidation would be unfavorable for the catalysis of malate dehydrogenase reaction to proceed in the oxidative direction. Knockout mutants lacking MDH showed that reaction proceeds in another direction, also in line with the conclusion, and clarified that oxaloacetate convert to malate and reoxidize the NADH produced by beta oxidation of fatty acid. This explains the compartmentalization is a mode of regulation. Next is the story of C4 photosynthetic pathway, another classical example of plant compartmentalization which is very tightly regulated. The regulation is tightly coordinated between mesophyll and bundle sheath cell as well as among chloroplast, cytosol and mitochondria. Carbon dioxide fixed initially in mesophyll cell cytoplasm by PEP carboxylase forms oxaloacetic acid, which further convert to malate or aspartate and is mobilized via plasmodesmata into bundle sheath cells. This will explain the role of enzyme compartmentalization as a mode of regulation again. Dear students, in this module, we have discussed how the complexity of metabolic pathways are regulated by enzymes, the main players, through compartmentalization. So to summarize, compartmentalization is a way of localization and regulation. Various methods of compartmentalization study include cell fractionation, immunohistochemistry, proteome analysis by mass spectroscopy, fluorescent protein tagging and other in vivo imaging, and finally computational or in silico approaches. Most of the primary metabolic pathways and their location have been well explored and mapped along with few novel pathways like glycolysis, glyoxylate and C4 photosynthesis. Thus, techniques in proteomics pave a way towards localizomics, where enzyme location was confirmed, which further studied as interactomics to explain the complexity of living system and its compartmentalization.